Hello, everyone, and welcome to today's webinar, Biofungicides, What Else Can Microbials Do for You? Presented by BioWorks. I'm Robin Sittberg of Meister Media Worldwide, and I'll be moderating the webinar today. This webinar has been approved by CCA for half credit in integrated pest management. To receive credit, you must watch the entire webinar. An attendance notification will be sent at some point during the webinar, and you must respond to receive the credit. If you respond, once the webinar is finished, you'll receive an email with a QR code for reporting your credit. You just simply scan the QR code and submit it. Please do not close your web browser before the webinar has ended completely, or you'll not receive the email with the QR code. So that's real important. I'll remind you again of that at the end. But this is also for the viewers watching the archived webinar as well. We'll have time for some questions at the end of the webinar. So if at any time during the presentation you'd like to ask a question, just go ahead and type it in your question pane and click submit. And you can do this at any time during the webinar, and we'll answer as many questions as we can at the end. Before we begin, I'd like to introduce our speaker. Dr. Michael Brownbridge is Senior Technical Services Manager with BioWorks. He provides technical support for the company's catalog of disease and pest management solutions and their application and integrated plant health management plans. Prior to joining BioWorks in 2019, Michael was fortunate enough to have traveled the world applying his craft with research and outreach positions in Israel, Kenya, the US, New Zealand, and Canada, putting new biological tools and knowledge into the hands of growers. So we have interesting information today that Michael will present, and so I'm going to turn it over to him.
Great. Thank you, Robin. I'm just going to switch over to the slides. Good afternoon, everybody. Thanks for joining us today. I'm happy to quote unquote see you, and we'll get started. I was interested. I've oftentimes talked about what else microbials can do, but never actually put together a whole presentation as a sort of a review of some of the benefits we can get from microbes, in addition perhaps to those we know as biological fungicides or biofungicides. So I just want to share some of that information with you and show you that a lot of microbials, they're just not one-trick ponies. They can do an awful lot that contributes in many, many ways to crop health and resilience. So when we talk about beneficial microbes, what are we actually talking about? Typically, we're referring to biofungicides, biostimulants, and microbial inoculants. Now, we know that plants are challenged by a range of biotic, that means living, pests and diseases, and abiotic stress factors such as extremes of temperature, drought, soil salinity, etc. And I think a lot of people are really in the midst of heat waves at the moment, myself not included. So plants are suffering a little bit. And effects of some of these abiotic stresses can be transient, and plants will recover just fine, but others, they have a lasting effect on plant performance. Abiotic stress also often renders plants more prone to biotic stress factors, meaning they're more susceptible to diseases and pests. And then if you look at the life of a plant, there are stressful periods in a plant's life too, when they're transplanted, when they're fruiting or flowering, for example. And this is where microbial products can help. They can enhance plant health and performance, which translates into improved crop quality and yields. Now, made an executive decision here that I'm actually going to focus on microbes in the root zone. And rather than me coming up with words to say why, I actually came across this recent article in CE Ag World, which I thought, wow, that's a great summary of the why. And I'll paraphrase. I know it's pretty small. I don't expect you to read that, but great article. If you have an opportunity, go in and read it. It's a really nice read. But it says, and I'm paraphrasing here, root systems are the engines of nutrient and water uptake, hormonal signaling and stress tolerance. And then when problems emerge, they often appear in the canopy, yet they are symptoms, not causes. So that speaks to the importance of root health. But it then goes on to discuss the influential physical and biological variables in the root zone, including microbial interactions and their importance to plant health and states. And the last sort of comment in the article is stronger roots mean healthier plants, more consistent yields, and better returns for every square foot. And that's why we're focusing on microbes that influence and benefit plant health through their interactions with the root zone. Now, microbial biofungicides used to protect plants against root diseases typically have two or more modes of action, protecting plants via a combination of competition through their growth habits, antagonism, their capacity to produce metabolites as they grow, which are can actually disrupt pathogen fungal cell membranes, bacterial cell membranes sometimes, but it also inhibits germination of growth of these fungi. They can induce plant resistance, and some, particularly those based on trichoderma, for example, can actually parasitize pathogens in the soil. So if we look at this, or the use of microbes within the context of the disease triangle, which summarizes what's required for plant pathogens to infect a host and spread within a crop. So it starts with having sufficient virulent pathogen present at high enough levels to infect healthy plants. The host plant must be susceptible, and environmental conditions must favor disease development. An infection really can only occur if all three of these elements are satisfied concurrently. That gives us nice insights, because if we can eliminate or reduce any one of them, we can significantly reduce the likelihood of disease developing and spreading. Of course, one of the ways in which we can reduce or manage pathogens is through the use of biological fungicides. And there's a range of biological fungicides that are available to you based on trichoderma fungi, products like Rootchill Plus, Asperello, and Optago, and bacterial products based on bacillus species, such as Cease, Rhapsody, Serenade, Stargus, Double Nickel, Serafel, others based on Streptomyces species like Lalstop K61 and Actinovate, and Howler EVO, which contains Pseudomonas chloraphis. So, back to the disease triangle. When plants are stressed, their performance is compromised, and invariably, stress renders them more susceptible to disease. So, stress impacts plant performance and can ultimately affect plant quality and yield, as I've already mentioned. And microbes can play an important role in moderating effects of stress through colonization of the rhizosphere, that's that thin layer of soil around plant roots, formation of biofilms along roots, production of compounds that promote nutrient uptake, and other important physiological, chemical, and biological activities that in turn invoke beneficial plant responses. And there's no shortage of data showing that Bacillus, Trichoderma, Streptomyces, and even Pseudomonas strains used as biofungicides and microbial inoculants have growth-promoting properties as well that help plants cope with and recover from abiotic stress events. They can stimulate root growth and development, proliferation of root hairs, which of course enhance a plant's ability to access water and nutrients. And they have the ability, through production of enzymes, to solubilize macro and micronutrients in the soil, making them more biologically available to plants, while concurrently enhancing their assimilation within the plant. It's quite amazing what a little microbe on a root can actually do. I find it totally fascinating. But then that begs the question, are all microbes created equal? And that's really like asking, are all humans the same? And we may all be the same species, but there is enormous variation in body makeup, physiology, and athletic ability. And we know that some humans are phenomenal athletes, while others, well, maybe not so much. The point being, there are differences too across microbial strains in terms of the benefits they can deliver. So you really want to use microbes that are Olympians, that have proven efficacy, resilience, and are great all-rounders, capable of protecting plants from soil-borne diseases and conferring other plant benefits. So my honest recommendation and advice would be, look for proven, registered, and reputable products rather than whatever's cheapest, because quality and performance count. So let's consider Streptomyces first. We're all familiar with it as a biofungicide, and it can bring a multitude of other plant health benefits as well, owing to its capacity to produce plant hormones such as indole acetic acid, or IAA, which promotes root and shoot growth. It can increase plant access to water and nutrients. Some strains produce enzymes that liberate and solubilize phosphorus from the soil, making it more biologically available to plants. Streptomyces, like most of the other organisms we'll talk about today, also produce and secrete siderophores. Now, siderophores, what a wonderful name. I always wondered what they were, so I made sure I had an explanation for you all today. Well, siderophores are actually ion chelating compounds that bind to insoluble ferric, that's iron 3, in the soil or in the environment, and then it forms a water-soluble iron 3 siderophore complex. That's recognized then by specific cell membrane receptors in the plant. And that complex can then be transported into the cell, where the iron is often reduced from ferric, iron 3, to ferrous, iron 2, making it available for various biological processes. And don't worry, there's going to be no chemistry test at the end of this show. And then last, some Streptomyces strains can fix atmospheric nitrogen, providing plants with obviously a key nutrient. And in this example, a Streptomyces strain with antagonistic activity against various diseases like Alternaria, Colototricum, Cladosporium, and Fusarium, and yes, I know some of those are foliar diseases, was shown to produce IAA. And treatment of pathogen-infected plants significantly reduced the onset of early blight and increased various agronomic traits in treated tomato seedlings. And the image clearly shows how the treated plants benefited compared to the control, with increased root and foliar biomass. And the plants responded to treatments containing whole cells, as well as the culture supernatant, showing that plant responses were primarily due, or in part due, to metabolites secreted by the bacteria. And then we see similar mechanisms associated with Bacillus strains. As they colonize roots, they produce a biofilm, which is an extracellular matrix of polysaccharides, complex carbohydrates, and other components that actually enable Bacillus species to multiply and establish a persistent association with plant roots. And that helps protect the plant from pathogens and environmental stresses like drought, and enhances nutrient absorption. It also facilitates the production of plant hormones, which further contribute to plant development. And we can see evidence of phytohormone production in that these plants were treated with Bacillus subtilis QST713, the active in Serenade, and the strawberry plugs were treated with the product prior to transplanting. And you can clearly see the growth benefits to the treated plants, which had stronger, healthier roots, which in turn supported enhanced foliar growth, resulting in higher yields per hectare. And I should say the plants began to yield faster than their non-treated counterparts. So some more data on Bacillus subtilis, the QST713 strain, this time in the form of cease, and my thanks to Dr. Michelle Jones at The Ohio State University for these data. And the trials were done to see how Bacillus subtilis could be used to improve abiotic stress tolerance. And this is particularly important when plants leave a greenhouse, either as finished plants or plugs or vegetable transplants. And there are many stresses on that journey, from propagation to production greenhouses, or from production greenhouses to retail. Plants can be subject to cold or heat events, water stress, low light conditions, and very often low nutrient levels over time. And what they found was that plants that had been treated with Bacillus subtilis in production held up better through transportation to the point of sale and recovered faster after these stress events. And that means less shrink at retail and better performing plants for customers. And some more data from Dr. Michelle Jones showing how microbial biostimulants can actually compensate for low nutrient levels. And in this trial, plants were fertilized with a mix containing 150 or 50 parts per million of nitrogen. And then a subgroup of the 50 part per million group were also treated with mycorrhizae as myco-apply or cease. And analyses of leaf tissue showed that nutrients like nitrogen, potassium, and others were higher in plants treated with a 50 part per million nitrogen fertilizer together with a biostimulant than plants treated with 150 parts per million alone. Cease also increased phosphorus availability, showing how these microorganisms can actually compensate for low nutrient levels in soils by helping release nutrients that may be locked into the soil or making them more accessible to plants. And the last example I'll show is for Bacillus specifically, speaks to how other plant health benefits provided by microbes impact the bottom line. And these are data from trials done utilizing a microbial inoculant in the form of the product Laurys Vita, which is based on Bacillus velezensis. And this was being used in the propagation of black cherry. Now, in the trial, the same growing medium, fertilizer, and watering regime were used in both treatments, but one series was treated with Laurys Vita, which was applied by drench beginning soon after seed germination. And there were a total of 3 applications made at monthly intervals. And in the results, you can start to see the benefits that resulted because of the Bacillus treatment. Namely, a significant increase in seedling caliper measures, accompanied by a significant increase in seedling root mass. And for the market these young whips are being sold into, their value is based on height and caliper measures, and different categories command different prices. Those with the best caliper, height, and root mass typically creates a more vigorous seedling for outplanting, and those seedlings are of higher value for the nursery. And indeed, that was the case. The increased growth of seedlings in the Laurys Vita program meant that the majority fell into the highest value category. And all the trees in the program were large enough to be sold. So significantly more income generated using Laurys Vita. And when you see, well, in the Laurys Vita program compared to the sort of conventional fertilizer program that the nursery had been using. And the calculated return was about $4 return on every $1 invested. So you start to see the added value that microbes can bring into nursery propagation and into other crops in terms of improved plant growth and potential for revenue generation. Other bacteria, particularly certain Pseudomonas strains, can also improve plant tolerance, particularly to drought stress. And this example, again, courtesy of Michelle Jones at OSU, the OSU. In this time, petunia plants were treated with Pseudomonas brassicarium. And these were more tolerant to drought than untreated plants. And in the trial, treated and untreated plants were not watered for 5 days. And both of these plants were just taken at the same time point within that trial. And you can clearly see the benefits that the bacterial provided to the plant on the right in terms of improved drought tolerance. In addition, bacteria-treated plants recovered faster when watering resumed. Now, many of the mechanisms induced or influenced by Pseudomonas species are similar to those seen with the other bacteria we've discussed, although it does have some unique traits as well. But as we look to the future, I think it's going to be increasingly important to use measures that enhance plant resilience in the face of weather extremes. Let's talk to trichoderma now. Now, trichoderma is a key component of agricultural soils, contributing to sustainable crop production through multiple mechanisms. Among its major roles, aside from suppression of plant pathogens, include the production of growth-promoting metabolites, production of volatile organic compounds, production of enzymes that solubilize soil nutrients and make them more biologically available to the plant, including phosphorus, potassium, iron, and nitrogen, and activation of plant immune responses, which enhance not only plant tolerance to diseases, but also to abiotic stresses. Now, trichoderma is well known for its ability to promote root growth. And this image was taken from one of several similar publications showing, in this case, how the volatiles produced by Trichoderma virens stimulated root growth. Not only stimulated root growth, but also lateral branching and root hair development in Arabidopsis seedlings. And we know that several commercialized strains of Trichoderma can do this. And this brings real-world benefits as well. And these images were taken at a commercial insectary producing celery plugs for field transplant. And the plug trays in both pictures were seeded at the same time. The same genetics was used, same growing medium, and the same watering and nutrient regime was applied to both. The only difference being that one set, the set to your right as you look at the two images, was treated with Trichoderma, in this case as Root Shield Plus, while the other was not. And you can see the difference in the level and synchronicity of germination in the root shield treated media and the more consistent and synchronous growth within that set of plants. And if you look at some of the plugs taken from those trays, you can clearly see how those treated with Trichoderma, Root Shield Plus, have more robust roots, the stems are thicker, the plants are just overall better. So, some real-world advantages caused by the use of Trichoderma. And as we know, transplants with well-developed root systems will establish or take faster. But there are other advantages as well. And particularly when we look at, as we increasingly move to more automated systems, particularly when we look at transplanting, a lot of transplanting is done what I'll call semi-automatically. There's still a lot of workers, but it's not individual workers putting plants down into the holes. But instead, the workers are feeding seedlings into that transplanter. Now, having a well-developed root system on the plug that gets fed down that chute is important because it helps the plug go down the chute and it will hit the furrow vertically. And it has to have the root system there, the well-developed root system, to hold together the soil because it sort of creates the weight that pulls the plant down and keeps it vertical inside the furrow. Now, this is with the semi-automated system. When we look at where a lot of field transplantation is going now, the plant tape system requires only 2 operators. Plants are grown in a bandolier-like arrangement of biodegradable cells. But again, to perform efficiently, the system requires high germination rates and a robust root system that holds the growing medium together in that cell so that the transplants plus the medium go through the transplanter and get planted into the soil without just falling apart in the process. And these pictures were taken in California, where Trichoderma hodsianum T22, and that's one of the actives in Root Shield Plus, was used as a seedling inoculant. And the pictures show how Trichoderma promoted synchronous germination and robust root growth in the lettuce seedlings planted or in the plant tape, making them ideal for transplanting. Plus, seedlings treated with Trichoderma are less prone to transplant shock, which further aids establishment. And of course, automated transplanters are not necessarily anything new in the indoor hort sector, but again, seedlings transplant better and take better when the root system is intact and holds the growing medium together through the transplanting process. Now, this image was taken at one of our customers that grows hellebores from TC plantlets. Now, the plantlets have no roots and they're basically just transplanted directly onto a Trichoderma amended peat medium. You can see how root growth was stimulated. And this is just 9 days after sticking the TC plantlet, with root growth that would normally take a few weeks. And that enabled faster turns within the greenhouse, increasing production efficiency through their ability to grow more plants in the same space over a year. And they've actually modified their production schedule accordingly to take advantage of this benefit. And really, no presentation would be complete without inclusion of a cannabis plant. And here, trichoderma applied at propagation improved root development in clones. And the trichoderma was only applied when those clones were stuck into those rooting cubes. Yet at harvest, the dry weight of cannabis flower was 4% higher from plants treated with trichoderma. And that's just one application. So not a bad ROI. And we see consistent improvements in rooting success in the presence of trichoderma. Both of these cases was Root Shield Plus, where granules were either incorporated into the growing mix or the wettable powder form was applied as a drench. And we see these rooting benefits in both woody and herbaceous perennials, here specifically in azalea and viburnum. And these pictures were taken at nurseries in Florida and Alabama by the growers themselves because they were really pleased with the rooting successes they'd seen through the use of these products. Now, these are production data taken from a commercial greenhouse in California, where the trichoderma-based soil inoculant RSSI was incorporated into the grow medium. And that led to increased propagation success of sedum. And the images on the left clearly show the growth benefits derived from using RSSI, much more consistent and lush growth, although you can't see it, better root structure and greater leaf area than in the standard growing medium. Plants were also more resilient in the face of abiotic stress and were ready for shipping much faster than was the case in the standard medium. And there was significantly less shrink in production. Now, what does all that translate to in terms of benefiting the bottom line? Well, sedum is typically grown on a 24-week growing cycle. And this was reduced to 20 weeks when RSSI was incorporated into the medium. So the crop had to be watered fewer times, resulting in labor savings of around $540 per crop cycle. And remember, these are just the savings from the reduced number of irrigation events. It doesn't include the financial benefits of reduced shrink, the added revenue opportunity through more efficient use of space and potentially labor. And then additional value comes from improved shelf life and other beneficial attributes at retail. So you can see how such products have the potential to deliver significant value at several stages in the production and sale of plants. I didn't know what else to call this section, so I just called it Crazy Other Benefits Derived from the Use of Microbes. When trichoderma colonizes plant roots, it will induce or prime plant defense mechanisms or plant defense pathways. So even though the applications are made to the roots, plants will mount a faster and more aggressive resistance response when challenged by foliar diseases. And in this case, a trial was run in tomatoes, and the disease was botrytis, and significantly more tolerant or resistant to botrytis than the untreated control in this case. And responses aren't limited solely to effects on foliar diseases. The induced responses, I kind of alluded to this earlier, can also have an indirect effect on plant pests. For example, spider mites develop more slowly on tomatoes treated with trichoderma, resulting in slower population growth. And aphid-infested tomato plants inoculated with trichoderma actually recruited higher numbers of parasitoids. I told you this was crazy stuff. And the specific parasitoid Ophidius urvi. So they recruited higher numbers of parasitoids due to enhanced production of plant volatiles. And those plant volatiles have been identified and are known to attract this particular aphid parasitoid, as I said, Ophidius urvi. Now, in both cases, I'm not claiming that the pests were controlled by the fungus, but the trichoderma did stimulate production of compounds by the plant in response to biotic stress events, and the observed effects on the pests have clear implications for improved biocontrol outcomes. Probably many of you familiar with Premier Tech's Promix mixes. Their Promix BS plus biofungicide actually contains both Bacillus pumilus, a biofungicide, and the mycorrhizal fungus Glomus intraradices. Now, fungus gnats are often problematic in propagation. The larvae can feed on young roots and fungi in the soil, and the adults can passively vector plant pathogens. And in research done in cyclamen, fungus gnat numbers were consistently lower in the Promix Plus treatment than in the standard peat-only medium. Inclusion of Bacillus pumilus in the potting mix appears to have a measurable and significant suppressive effect on fungus gnats. Now, why would that be the case? Well, fungus gnats, the name kind of gives it away, I think. They preferentially lay their eggs on the hyphae of phytopathogenic fungi, such as Botrytis and Fusarium, and they'll feed on these and other fungi in the substrate. As Bacillus pumilus suppresses fungi in the growing medium, it may affect the quality and quantity of food available to the fungus gnat larvae, and make it less attractive a medium for the adult fungus gnats to oviposit into. And I suspect this is something we would see with other biofungicides as well, which do have this effect on fungi in the soil. Now, a common question I get, and I know comes up a lot in this sort of presentation, is can I use microbial products together? What about compatibility between, say, microbial biofungicides and other beneficial species like mycorrhizae? Short answer, for most Trichoderma and Bacillus species, yes, absolutely. They can be effectively used together with mycorrhizae. And in fact, research, as I've shown in the slide here, suggests that they typically complement each other in the soil, with plants benefiting from the services that both organisms can deliver. So what about compatibility between Bacillus subtilis, the QST713 strain in cease, and Trichoderma as root shield plus? Now, when both organisms are co-inoculated onto an agar medium, the bacillus can inhibit growth of Trichoderma, but media is not the soil. What actually happens in the soil? So to test this, cease and root shield plus were applied to lettuce seedlings in 3 different regimes. Cease was applied 24 hours before root shield plus. It was co-applied with root shield plus, or 24 hours after root shield plus. And then root colonization by Trichoderma was determined 5, 10, and 15 days later. Now, root colonization values are inherently variable. It's just the noise in the methodology that we use. But if you look at the trends rather than the actual numbers, you can see that colonization levels in each treatment are actually relatively consistent over time. The inference here is that root shield plus is compatible with cease. And then we have quite a few other studies that have been done, similarly showing that Bacillus species often co-occur in the rhizosphere with Trichoderma. And Trichoderma, we have an increasing sense, seems to have a very strong influence on the makeup of the microbiome. Now, some publications actually indicate synergy between the two in terms of improved disease control and plant performance. And I picked out a quote from one of these articles regarding use of Bacillus and Trichoderma, which in essence states that their combination offers even greater potential as plant growth promoters and as biocontrol agents. Synergies between Trichoderma and bacteria cause more benefits than the sum of their parts. Such is the joy of synergy. And continuing with Bacillus and Trichoderma, these are compatibility data from Impello Biosciences, who showed that there was no growth inhibition of Trichoderma hartzianum, the T22 strain, when co-inoculated onto plates with the Bacillus strains contained in Impello's Tribus and Continuum microbial soil inoculant products, indicating that these species will co-colonize the rhizosphere and there's likely synergy between these organisms in terms of improved disease control, plant growth promotion, and resilience. So if you aren't convinced yet about the benefits that microbes can bring you, I do hope you are, because I truly believe that they have an awful lot to contribute in horticulture in general. The question often becomes as well, sometimes I think, what about the organisms that are already in the soil, or what can I do to enhance the microbials that I'm applying to the soil? Because we know that in soils with low organic matter content, low carbon levels can be a limiting factor to the establishment and performance of beneficial microbes like Trichoderma, Bacillus, even Pseudomonas. And in such cases, it can be beneficial to co-apply them together with materials that provide a source of carbon. And I know many growers use simple sugars such as molasses, but these are really also readily utilized by other soil microbes, not all of which are desirable. And they often induce a very rapid spike in non-spore forming bacteria. So are you truly accomplishing what you're seeking to gain from this application? I would preferentially like to see use of materials which are less accessible to some of those bacteria, such as protein hydrolysates, commercial products like On Guard and Trainer. And as you can see from this image to the right, the growth of Trichoderma hartzianum is as good on On Guard amended water agar, so On Guard was the only food there, as standard PDA, which contains protein and sugars as a food source for the fungi. And there's field evidence to show that protein hydrolysate promotes Trichoderma growth in the root zone of plants treated with the fungus. And then similarly, microalgae and products like Phycoterra can actually be utilized by Trichoderma, Bacillus, and Pseudomonas, as these images show, with dramatically enhanced growth of Trichoderma on soil agar amended with the product. And other biostimulants such as seaweed extract, humic acids, chitosan also promote microbial activity in the rhizosphere and are highly complementary to Bacillus, Streptomyces, and Trichoderma, either in the native microbiome or following their application. So, biofungicides, biostimulants, and microbial amendments are versatile, multifunctional products. I believe excellent additions in an integrated plant health management program. Biofungicides have proven efficacy. They bring unique modes of action to a program, which is essential for resistance management, and they can elevate plant growth performance. The short REIs mean they can be applied with minimal disruption to day-to-day crop management activities. And then biostimulants bring additional advantages to plant health and resilience. And use of these materials together can help address some of the biggest challenges facing the horticultural sector today in a very cost-effective way. And with that, I'm going to conclude. It's time. You've had enough of me. So, Robin, if we can open it up to questions now, I'd be happy to take any.
Absolutely. Thank you for that presentation. We've gotten a number of questions in already, and we'll start with those. But if you have a question you haven't submitted yet, there's still time. We've got about 20 minutes to answer some questions. So our first one is, let's see, can cease provide the same level of drought resistance if it is only applied once on plants likely to encounter drought, or would multiple applications be a better practice?
That's a darn good question. I think, as with many of these microbes, you get the best benefit from the microbe by ensuring that there is sufficient colonization of the whole root system. And plants obviously grow, their root systems will expand, particularly early on in the plant's life. So I think it's important to repeat applications of microbials like cease to ensure that you always have sufficient microbes there to colonize new roots, which may not have been contacted by that original application. Because even though the microbes will grow on and along the roots, I think the roots can grow faster than the microbes can colonize them. So I would say for best benefits, particularly in those early stages of growth, to get the best protection you can of the roots, you should make more than one application. That doesn't mean you have to apply throughout the entire life of the plant, but certainly during those early growth stages.
This is my question now, what would a recommended interval be for those multiple applications, do you think? Or does it depend on it?
Yeah, I think that's situation dependent, to be absolutely honest, Robin, and it depends on the soil, it depends on the location. I cast my mind to trials that one of my colleagues did in strawberries in California, and they were applying Root Shield Plus, the trichoderma in this case, to the soil together with On Guard. Now, and I'll give you the context for this, why the applications mattered where they did. The soil itself was basically devoid of organic matter, had high levels of fusarium. It was hot. There were, everything you could have that was bad in the soil, they kind of had it there. So in order to get the trichoderma to establish, they needed to inoculate the roots quite frequently. And they applied it together with a protein hydrolysate product to promote colonization of the roots. But they were applying every 2 weeks there. Low organic matter soils. Higher organic matter soils or growing media, peat-based media, coir-based media, you probably don't need to apply more often than 4, 6, 8 weeks. Then it becomes an issue of how fast is your plant growing, perhaps, or what are your disease pressures. And sometimes, if you push it too far, trichoderma or bacillus don't just disappear, but their numbers decline over time in that medium. So you're really just topping up. What I would say is that first application is probably going to be the highest level you will use. Subsequent applications, you can probably cut that application down by half. Certainly we know with the trichoderma, say we're going out at 8 ounces per 100 gallons, we can cut that down to 4 ounces per 100 gallons for subsequent applications. But application interval and level, you have to sort of understand your situation. What are your disease pressures? What's in your soil? So it's not a simple, just do this and it'll work every single time.
Yeah.
If only life was that simple. But so,
Right.
so there's caveats to every single response I've given. But I think I've covered the ground to give people an idea of what they need to be looking at in order to make that decision.
Okay. All right, our next question is, are there temperature limitations on trichoderma? For example, will it be effective to drench during 100-degree weather?
Yeah, that's a really good question. I don't think anything's effective in 100-degree weather. But let me add this. I'm certainly not effective in 100-degree weather. But is it 100 degrees ambient temperature or is it 100 degrees soil temperature? It makes a big difference. I think if you look at any plant in that type of condition is probably going to be watered quite frequently, and any irrigation event is going to cool down that growing medium below that 100-degree ambient. Is it ideal? Absolutely not. Would I plan to make all my applications at midday on a 100-degree day? Probably not. But 100 degrees in itself is not necessarily a death sentence for any of these microbials. It means that there may be temperature extremes that the trichoderma just shuts down. It will tolerate temperatures up into the high 90s. So it may not be growing. It just sort of becomes quiescent. It just shuts down and then waits for temperature conditions to be more favorable for the trichoderma, let's say, to grow. Similarly with bacillus. So my great answer to that question, it depends. But there you go.
Right. All right. We have a question where someone wants you to give the same talk to one of their groups, so they obviously liked it.
Yes, Sebastian. Of course I will, Sebastian.
How about this next question is, are biofungicides compatible to be tank mixed with PGRs?
Ooh, yeah, I think so. I'm going to have to put hand on heart, say, I don't know. I would have to consult our compatibility guide for that. We know that many products can be compatibly tank mixed with many fungicides. And I'm sorry, I can't give you a direct answer. I would refer you to, certainly on the BioWorks website, we have a lot of compatibility data available for our products, as I say, between Trichoderma, Bacillus, and other living microbes and other products that we have, and other synthetic products and biological products. But also, I know there's some good information available, say, on the Lalamand website around compatibility of their products with other products too. So, I apologize, I cannot give you a quick off-the-cuff answer to that one. I too would go to those sites and check out the information that's available.
Okay. We have a question about how strep, BT, trichoderma, etc., can potentially affect the composition of indigenous soil microbial communities.
Oh, yes. Am I still there?
Yes. Whoops. We lost him. Dial back in, Michael. He'll be back, I'm sure. Not sure if he just accidentally closed out but he'll be back.
Sorry, Robin. I dropped off then. I apologize. That was
that's okay.
I was just trying to click down the list of questions.
Yeah, the question is, can any of these microbials that you've talked about on the webinar potentially affect the composition of indigenous soil microbial communities?
Yes, they can. And I think the one we perhaps know most about is trichoderma. It will influence the composition of the soil microbial communities. It certainly, and I don't want to anthropomorphize trichoderma, and say it allows. You know, but it does. Trichoderma present will result in a decrease of certain microbes, oftentimes the less desirables. And facilitate, enable populations certainly of Bacillus. It plays very, very well with trichoderma in the soil, it seems. Streptomyces too will play really well with trichoderma. So for sure, they will influence the native microbiome, for sure, to a point. But you're shifting the population dynamics in favor of beneficial species and away from less or detrimental species. And I would say, soil is very, very resilient. That shift will change again as the plant gets older, different stages in its life. It will recruit in different microbes and making them. The plant itself probably has the biggest influence on what goes on in the microbiome. So it will change because of the plant. And once the plant's died, the microbiome will shift again. Soil is a pretty resilient thing if you just leave it alone to do its thing, or just try and influence it a little bit one way or another. But I'd be lying if I say that there wasn't a potential influence, but I think it's in the positive direction, not in the negative direction.
Okay. I have a question here about, it says at the beginning of the webinar, you mentioned you'd be talking about beneficial microbes in biofungicides, biostimulants, and microbial inoculants. Don't they all do the same thing?
Oh, what a perfect question, Robin. Again, let me give you this great answer. Yes and no. Microbial biofungicides, if I focus on them, they are microbes which have somewhat unique capabilities in that they have known mechanisms by which they can suppress growth infection by plant pathogens. And they've been commercialized based on that capacity to induce plant responses, actually parasitize pathogens, or their production of certain compounds as they grow will inhibit pathogens, or just because they colonize plant roots. They take up all the space and nutrients, and they don't allow competing pathogens to sort of move into that space. That's a somewhat unique feature to some Trichoderma, Streptomyces, Bacillus, etc. strains. Now, some of, backtrack, biofungicides I think pretty well every single one of them also has biostimulant properties, in that they can solubilize nutrients in the soil, make them biologically available to the plant, and enhance uptake and assimilation. They can induce plant defense responses, as I said, but the broader context in terms of allowing the plant to cope better with biotic and abiotic stress factors. Now, some of the other microbes, they may be Bacillus streptomyces, trichoderma, don't have the capacity to mitigate plant diseases, but they do have the capacity to solubilize nutrients in the soil. And that's perhaps why they're used. So then they're not all the same. A lot of them have the same name, Bacillus, streptomyces or whatever, but not all strains are the same. That's the whole point of including, the strapping athlete and the couch surfer. So I think it's important to acknowledge that and recognize that. And I all right, hand on heart, pet peeve is seeing something that says contains 20 different microbes. It's like, well, so what? What do they do in the soil? Do you think they all coexist? Are they all viable at the time of purchase? Do they actually perform once you've applied them to the soil? Are they at meaningful enough levels, meaning that any microbe applied to the soil, I mean, theoretically, you could apply 10 and they would ultimately colonize the roots. But how long will that take? So by using higher numbers, you're facilitating faster colonization of the roots. All right. Yeah. We know that over time, those 20 microbes, there may be two, maybe 3, which will ultimately survive to colonize the rhizosphere. So I think it's a somewhat disingenuous claim around more is better. It isn't. Having the right microbes in the right combination potentially is what matters. It's not just having the number. It's the microbes and the level of inoculum that you apply to the soil. So back to your question in this sense, it's important to, I think I said it, no, they're not all the same, but I think it's important to understand the different products. There's a lot of products I know out there that they're basically selling dust. There is nothing living in the bag. We know that for a fact. So reputable products, they may be a little bit more expensive, but at least you know what you're buying and you can guarantee what it says on the label is what you're purchasing. And when you're looking to use that microbe for plant health purposes or for plant enhancing plants' resilience to abiotic stress, that's pretty darn important.
Okay, well, this next question is sort of in line. It'll sort of a follow-up to what you just said. Can you talk about the importance of a quality formulation to enhance the efficacy of microbials?
Absolutely, yeah.
And I'll just say, and how would you know, if you're just out there looking at a shelf or talking to somebody, how do you know if they're good quality and the right formulation?
I think some of that comes down to who do you trust and who's got the data to back up their product in terms of showing how well it can perform. There is no shortage of interesting and high-performing microbes out there. Where the difference lies, in many respects, is how you produce that microbe. So production is important, and consistency of production, such that at the end of it, you produce a very stable spore. Can you hear me? Can you still hear me okay, Robin?
Yes.
Okay. But it then comes of it's no good just to have that spore. It's how you preserve it. And that's in part what the formulation does. It will preserve that microbe in the jugs or the package, so to speak, so that it will remain viable from point of production and formulation to the point of use. And then many, many formulations contain other ingredients which actually improve the efficacy of that microbe when it reaches the plant. And particularly for foliar microbes, the formulations there do include a lot of adjuvants which help enhance sort of leaf spreading, leaf wetting, even potentially persistence on the leaf, but also in the soil. What other materials are used in the formulation to improve ease of use, by way of ease of mixing. How easily does a wettable powder go into suspension? How easily does a liquid formulation mix with water? You know, if it's an oil formulation, oil floats on water. But good formulations, actually, they contain other materials that the product may be pre-formulated that really enables really good mixing. So you get a very homogeneous mix of that product. So formulation is almost 80% of the game when it comes to efficacy. The microbe method of production, formulation is the real piece you need to get it into the right place. I should say, and I forgot totally, application matters too. Between those four things, those are the key four. But application, ease of application is often facilitated by formulation. So they're all linked, all subtly different, but all important.
Yeah, yeah, good. Yeah, I'm glad that question came up because I know, I mean, formulation is important with anything you apply, but certainly with microbials because they're alive.
Yeah.
Let's see. What are some of the common mistakes you see in reducing the efficacy of microbial products? The opposite of what you just said.
Opposite of what I just said. Yeah. Play that in reverse and you'll get there. I think tank mixing with the wrong material. The two have to be compatible. Check compatibility before you tank mix, not after you tank mix. And that goes, you know, we talk about compatibility being physically compatible, which speaks, formulation comes here too, because sometimes formulations are incompatible in that they'll get globs of, do you remember those lava lamps from the 70s? You're way too young to remember that, I know, but the gloopy stuff that went around in those lava lamps. Anyway, you can get stuff like that. You can get solids form or materials just drop out of suspension or solution. So you have to be real chemical, important. That's physical compatibility. Obviously, with living products, you want biological compatibility as well. And that's where tank mixing with certain chemistries is absolutely fine. With some chemistries, absolutely no. So that's a common mistake. And then using product beyond the expiration date, I think, is another good one. And then using living product which has been stored in a shed at I'll go higher, 120 degrees in the summer, and expecting it to be just fine when it's pulled out and used it. So those are probably the three common ones. Okay.
Got a couple more minutes. So one more question is, again, it's kind of following up with it. What is their optimum pH, both in the soil for applying any of these products and also in the tank mix? Is there an optimal water pH when you're mixing?
You want to be ideally in that sort of, I'll say, 5 to 7.5 range, which is around neutral pH. Organisms like trichoderma will function in soils down to pH of about 4, up to about pH of about 8. So you have a wide pH range at which you can, within which you can play. So, but every microbe has its optimal. You know, the optimal for trichoderma is in, it's about 6 to about 7.5, between that range. But it will function a little bit slower, but it will still function just fine at a broader pH range. In the tank, again, as long as you are within that range, I think you'd be fine. You don't want to be putting anything into anything that's really acidic or really basic.
Okay. All right, good. Well, I think we're about out of time. We're exactly on the hour. So I want to thank you, Michael, for doing this. It was great, really informative. If anybody wants to watch this webinar again, it will be recorded, or it has been recorded, and you can access it at the same link that you used to log on today. So you can go back and screenshot it, or rewatch parts of it, or the whole thing if you want. And you'll get an email in your inbox tomorrow that'll remind you of the link, so you can go on and do that. As a reminder for those who responded to the attendance notification for the CEU credit, remember not to close your browser before the webinar is completely ended, or you won't get the email with your QR code. And again, this is for viewers watching the archived webinar. So With that, I will close out and thank you all for attending, and thank you, Michael, and thank you to BioWorks for making the webinar possible.
Thanks, Robin.
You're welcome.